Penguins. So the FRQs from Thursday have dropped, and so it was asked for me to go over the answers that I have. So I don't have the fancy little PowerPoint that I usually have, but here is the Word document that I created to share with my students. I do want to remind you that I do not have any scoring guidelines. I do not have any insider information. These are just what I would have written if I was sitting for the exam on Thursday, just to give you an idea of different answers. That doesn't mean that your answer is incorrect. There could be additional scoreable answers to these that would potentially be available for you to have scored points. And I also could be wrong on some stuff. Okay. So I'm going to run through all 6 of them because I was asked to just do one straight video. So I hope that this is helpful. So the first question talks about crossing over, and that's required for homologous chromosomes properly aligned during metaphase. So this is important because we need to make sure that we understand that the crossing over is what's going to allow for them to actually go through that law of segregation during anaphase. So A asks us to describe the function of the S phase. S phase is during interphase, is right after G1, and that's when our DNA is going to replicate. So we make a copy of that DNA strand, so we'll be able to have equal distribution of chromosomes during nuclear division. It then goes through this whole big long explanation about hotspots, and it talks about that there's a higher frequency of crossing over in those regions. They have different genes. There's a red fluorescence protein as well as a green fluorescence protein, and they're seeing what happens after it crosses over. So it crosses over. We see that only the 2 inside ones are going through that crossing over process. So we have one chromatid that has the red protein, one that has none of the fluorescence, and then one that has both green and red, and then one that just has green. So it says to explain why haploid cells formed after meiosis will only have one of the markers. And so I said that it had to do with that these 2 kind of interior non-sister chromatids were the ones that were going to exchange that genetic information with crossing over. And so it would lead to you having the 4 different gametes, one that has each of these different chromatids. So these 4 different chromatids would be separated doing meiosis 1 and meiosis 2. That would lead to these 4 different options inside of the cells. So they then investigate that there's a certain protein. They want to look at the kinetochore protein. So they have 3 groups. Group 1 has no kinetochore proteins. Group 2 has a CTF attached to the hotspot. And then group 3 has an IML attached to the hotspot. Okay. And so they give us this graph. They're showing us the frequency of crossing over with these 3 different proteins. And they want us to get a control group. Control group is going to be our first group that had no kinetochore proteins attached to it because of the fact that it's not going to have, we're seeing how these different proteins affect it. And so not having the proteins, what we're going to use to compare. We then have to say, okay, well, they followed it up with a modified version of the CTF and it had been removed. And then they were testing the presence and absence of unmodified, as well as the presence and absence of the modified. And they didn't give us the data, but they wanted us to justify the use of that modified protein. Okay. And so I talked about how it would be unable to bind to the DNA. And so they're going to be able to determine whether it was the CTF, or if it was something about the binding site. So they were going to be able to really determine what was the cause for the decrease. Because if you look at the data, there was a decrease with the CTF and that frequency of crossing over when that chemical was used or that kinetochore protein was used. And then we have to identify an independent variable. And so the independent variable I had was the presence or absence of that CTF protein in the modified or unmodified format. Part C says describe the effect of the frequency of the crossing over when it's attached to it compared to when this other one was attached. And so we see that the crossing over percent decreases because we can kind of compare that to the control. So when you compare it, you can see that, it's not going to zoom, sorry. So you can see that the error bars overlap when you compare the CT, when the IML is compared to the control, you see those error bars overlap. So this one is going to show us no effect, but the error bars between the CTF and the control do not overlap. So we can say that it actually decreases that, and then we have the effect of that one. We did see, okay. So D, we want to predict the effect of the number of copies likely to be present in the resulting cell. And so we do see there is a decrease when you have the CTF. And so I talked about that since we had the decreasing of that recombination, the decreasing of the crossing over, that's going to lead to a decreasing of these homologous chromosomes from being able to segregate. And that's going to lead to non-disjunction. So there'll be additional chromatids in some of the cells, and they'll be missing a chromatid in other cells. And so the justification was that, of course, homologous chromosomes are unable to segregate during meiosis if we are unable to cross over, and that leads to that non-disjunction, which gives us the incorrect number of cells. And then part B3, it talks about the presence of these hotspots, increased likelihood the population will survive in the presence of selective pressures. Okay, and so having the hotspot, it increased the amount of crossing over. It had the high frequency of crossover. Crossing over is going to reshuffle the genetic information. It's going to give you new combinations. And so those new combinations could allow for an individual to be more favorable. And so once that individual is more favorable, we're going to see the population is going to kind of change and have that favorable trait. So that was number one. On to two, we're investigating the increases in environmental temperatures affecting the metabolism of different organisms. They look at liver toad cells, and they look at these different markers. The first thing was describing the role of water in hydrolysis of ATP. So hydrolysis was just breaking that water, the bond that's in the water, and then you use the hydroxyl going to one side and the hydrogen going to the other to break that bond between that the third phosphate and then the ADP. So that we're able to break that bond. So B wanted us to make a template, like we were using a template of the graph. And so the information I was provided was that it was a 20 by 30 graph. So that's what I tried to do for y'all. So it told us that we're constructing a bar graph. So you should have made a bar graph. And so I think that you would only really need to label the one y-axis. I do see why there could be a double y-axis on this, but in terms of the fact that these are going through the same numbers and they're both looking at rates, they have the exact same units. I use a single y-axis on it. Okay. And so here is my graph. So I've got each of the points plotted. I've got the error bars plotted. And I use the key to differentiate between the oxygen consumption and the ATP synthesis. And so the way that I separated them was I kind of like made a hash mark in one of them and no hash mark on the other. And then I counted every 2 blocks was 1. So I went up by 0.5. So that would be allowing me to get to the full number. And then I started the graph at 10, and then I had each of the temperatures. I was too lazy to look up the symbol, so you just have a little asterisk. So just deal with that. Okay, cool. And then you labeled your axes. And then part B wanted us to determine the temperature at which the rate of the oxygen consumption is different from 25 degrees. So if I look at my 25 degrees, I can see that 20 and 25 at oxygen consumption, their error bars overlap, which means that that is not significant. But if I look at 30 and 25, I can see that those error bars do not overlap. So I would say that the temperature was 30. So 30 has a different rate. Then C wants us to describe the effect of the temperature on the rate of ATP synthesis. Okay. And so you can kind of see that at lower temperatures, our ATP synthesis is low. And as we increase in temperature, our ATP synthesis increases. So I said that it was a direct relationship because as temperature increases, rate of ATP synthesis also increases. So I've had like a little mental, I can't figure this out in terms of the math. So like I've always was taught that whenever you had like a fraction in the denominator, that you would then like flip the fraction and do the reciprocal of it. And so the answer I have here is kind of what I originally was thinking through, but then I also could see it being a different answer. So I really don't know what the right answer is here. But so I've got 16.5 nanomoles times 10 minutes, and that will get rid of the minutes that's in the denominator, leaving me with 165 nanomoles per milligram. And I did the same thing, multiplied by 10 again, which gives me 1650 nanomoles to be the number. Okay, I could see where you would potentially flip it. And if you did that, I believe the 10s are going to cancel each other out, which will just leave you with 16.5 nanomoles. Honestly, I could just be overthinking it because I'm kind of tired from, everything. But so the answer is either going to be 1650 or it's going to be 16.5. I think the answer is probably 1650 and that I'm overthinking this. So then part D tells us that there's this compound that blocks the chemical function of a protein and ATP synthase. So we want to predict the effects of that on the proton gradient. And so I talked about how the proton gradient is going to increase inside of the intermembrane space because of the fact that you're blocking the channel from allowing the protons to flow through it, which is also going to cause there to be a decrease in the pH because there's that buildup there. I did talk about ATP just because that's just where my brain was going, but I think it really needs to focus on with the protons being stuck in the intermembrane space, and so they're unable to get back into the matrix because of the fact that that protein is inhibited. Number 3, we want to investigate red blood cells and their ability to take in glucose from their environment as they age. And so they use different ones. They had a day 1 versus a 7-month-old. And then they use radioactively labeled glucose to compare them. So we want to talk about the difference between passive and active transport. Passive requires no ATP input versus active transport requires that ATP. Passive is going down its concentration gradient, while active is going against its gradient. So I'm not really sure where they were wanting for that. They probably would accept both of those answers. So justify why the sensitivities use equal numbers. So you want to use the equal numbers so you'll be able to eliminate the fact that it could be the number of cells that's leading to a difference. So we have the exact same number of cells is going to be considered a constant in our experiment. So it allows us to determine, was it like actually the glucose, not the number of cells. C talks about the glucose transporters are required for moving of that glucose into the red blood cells. So they claim that there's a certain gene, and that gene is going to decrease in terms of the number of transporters as those guinea pigs age. So the claim is then we want to predict how the increased age on the amount of glucose. Well, if we're saying we're not having as many of those transporters as guinea pigs age, we're going to see that as the cell increases, the expression decreases, so we'll see less of those radioactively labeled glucose molecules in our older cells than we do in our younger cells. And of course, justification has to do with the fact that looking at the number of cells and the amount of expression that was taking place in it. So if you have less expression of the gene, you're going to have less of the protein, and so we're not going to be able to bring in the glucose as readily. So part D is our ecology question. This one's talking about the wild oats is native in Europe and Asia. There's an invasive species in Central California. So we see that this common wild oat is replacing the native bunch grass. There's also this aphids that are these little insects that carry different viruses. And so we're kind of looking at that. So part A describes the change in the resilience of an ecosystem when there's a decrease in the number of species. And so if we're looking at that decreasing in species, that means I'm going to see a decrease in my resiliency because I'm decreasing my biodiversity. And so the less of the diversity you have in that community is then going to, of course, lead to a decrease in that resiliency to kind of resist changes. So part B asks us to explain the addition of the common wild oat affecting these bunchgrass plants, and then how to be supported by the grassland. So wild oat is an invasive species, and so it's replacing many of the native bunch grasses. So there'll be a decrease in that bunch grass population, and then the aphids are more likely to affect the bunch grass, which also leads to a decrease in their population. So we're seeing how that is affecting those bunch grasses. And then part C asks us to suggest, like we use ladybugs, which will eat the aphids, predict the effect. So if the ladybugs are eating the aphids and there's less aphids around, they're going to carry that virus that will infect the bunch grass. And so we're going to see less of the viral transmission. So we should see an increase in our bunch grass population because less of them will be infected and less of them are going to die from the viruses. Part B has studying mechanisms that enable or prevent speciation, so a post-zygotic mechanism that prevents our gene flow and thus enables speciation. There's 3 different post-zygotic mechanisms. We have the reduced hybrid viability, we have the reduced hybrid fertility, and then we also have the hybrid breakdown. I think if you named any of those 3, you would have been fine. It does say to describe, so you want to give more than just a name drop. So just describe what those are. So then talk about these different regions of the DNA, and then they study that there's this antifreeze glycoprotein that they found in some fish species. It reduces the freezing temperature for the fish, and so that's this AG gene that we're looking at. They then gave us a phylogenetic tree, and yes, I was wrong. I didn't think they were going to do phylogenetic tree 2 years in a row on the same question, but here they did. So They then asked us to explain how change in the genome enabled the cause to survive and reproduce after a period of freezing temperatures. And so right here is where we see that freezing temperature. You can see that before that freezing temperature, there weren't any of those AG repeat sequences. And afterward, we see that they're around. So there must have been a mutation that led to this repeat sequence. And because of the freezing temperatures, those individuals were more viable. And so we're going to find that they're more likely to survive, and they, of course, pass on those traits to the next generation. And so it kind of emerged during that time, and then the selective advantage of that they could have a lower temperature than the other fish allowed them to be selected for in that environment. Part C asks us to use the template to place an X to represent the origin of that gene. So I put it on this point right here because before that point, we did not have any of that gene. And so if you kind of go back to so it has to have come from this branch right here that led to where all of them are. And I've now lost the diagram because I try to scroll in. Okay. Then part D says to explain how the genetic differences among the family determine habitats in which they live. And so you kind of see that those that lost their trait are in warmer temperatures as well as the ones that never had them. And then those that have that AG trait are going to be in the colder environments. And so we can kind of see that those that have the allele are more favorable in those environments and can survive in those environments. And I'm assuming they probably have more food than if they were to try to go into another environment where other fish are. And so they have kind of that like small little niche that other individuals can't compete with them in there. And so they're more likely to survive, and they're more likely to leave those more offspring. And then 6 talks about that we can quantify the rate of translation as ribosomes move along mRNA from one codon to the next. So what they're doing is they're looking at the rate that these mRNAs are translated. And so they're looking at also the waiting rate of it. So they're seeing the number of times the rate was recorded, as well as the translation rate of those different genes. Okay. So A asks us to identify the rate was recorded has the greatest number of times for GAC codon. So if you look at the GAC codon, you can see that there's the highest point right here at 150. So I think it's 150 ms per codon. And then part B says the graph B and C describe the variation in translation rate of AUU codon compared with that of the UGG. And so when we look at this, we can kind of see that there's a higher amount of translation at the beginning for the AUU versus the UGG kind of stretches out a little bit more, and it takes a little longer for it to get started, and it's a lot more flatlined. And so that could be a difference in the available codons. So the high number of rate of translation is low when UGG has a lower number of times. It kind of shows that inverse. And then I just made like a weird little wonder there. So part C, scientists hypothesize that molecules that bind to UGG codons are available at lower abundance than the ones that bind to AUU, so we want to support it. So UGG takes a little longer to get started with the process, and so it might be waiting for those available codons to come about, which might be why you see the kind of the slower increase that take place with those. And then part D, amino acids can be encoded by multiple codons. Some of them can have the same amino acid occur more frequently in mRNA than other codons. Explain why the use of one codon over another for the same amino acid might result in increased levels of protein production. So if you have more of these different codons, that means that you're going to be able to bring in more of those amino acids so you can increase the rate of that translation. So I hope